Thank you once again for inviting me, and I'm very, very pleased to participate in this conference as a keynote speaker. I would like to talk about the use of some sequences of chloroplast DNA in rye species. The topic of my presentation concerns the use of non-coding regions for analysis of rye species. Cultivated rye is an important cereal in Northern and Eastern Europe. All species within the genus Secale are diploid with 14 chromosomes. They are easy to cross with each other and are formed partly by crossing fertile hybrids. Only Secale sylvestre has different characteristics and is probably the most distant species. Rye is a rich and important source of viable genes, encoding, for example, high protein content, disease resistance, as well as morphological and biochemical traits, enhancing the value of triticale. In the wild species, for example, Secale wawiowi or Secale sylvestre, disease resistance genes are sought, for example, powdery mildew and leaf rust, resistance to pre-harvest sprouting and lodging. For breeding cultivars with increased grain protein content and a variety of assigned green mass, Secale anatolicum is used. Wild species of rye are also a source of male sterility. Despite the economic importance of rye and a large number of existing studies, the taxonomy of genus Secale has long been a subject of many discussions and debates. Some of these studies suggest a division of genus Secale into about 14 different species. However, Fredrickson and Petersen recognize only 3 Secale species: Secale sylvestre, Secale strictum, and Secale cereale. According to the classification system adopted by the American Germplasm Resources Information Network, also used in this study, genus Secale now includes 4 species: the annual Secale cereale, Secale sylvestre, Secale wawiowi, as well as the perennial Secale strictum. Moreover, Secale cereale includes 8 subspecies, the Secale strictum 5 subspecies, while Secale cereale subspecies cereale is the only cultivated species. All these studies do not explain fully and clearly the evolution of rye, perhaps because the genus is relatively evolutionarily young when compared to wheat and barley. Determination of phylogenetic relationship among taxa of wild and cultivated species is extremely important for the process of breeding new cultivars, because it will facilitate transfer of the genes encoding these traits to the cultivated species. The differences in the classification of genus Secale results from the application of various test methods, such as ITS, this is internal transcript spacers, or macrosatellite analysis. Some of these studies also include the organellar genomes. Although plastic DNA has been widely exploited as a marker of choice for phylogeny and phylogeography studies, little is known about its utility for examining relationships between closely related species. The slow rate of evolutionary changes inherent to chloroplast DNA make it difficult to perform lower level taxonomic analysis, particularly at the population level, which is neglected in lots of studies. In plants, non-coding chloroplast DNA regions have been explored for lower level taxonomic studies because they are under less selective pressure than coding regions and show an amount of polymorphic site useful for phylogenetic studies. However, the phylogenetic signal of different non-coding chloroplast DNA regions can vary extensively among taxonomic groups, and the rate of evolutionary changes of different regions are poorly understood. The coding regions of chloroplast genomes of higher plants are highly conserved across species and genera. However, the non-coding regions of the chloroplast genome have been described as highly variable. This has led to design of parts of universal primers which can amplify non-coding regions. Because these studies have not been made on species of rye, we decided to check the usefulness of these universal primers. Our aim was to test whether the published universal primers of chloroplast DNA could amplify respective non-coding regions in Secale species, detect interspecific variation in the chloroplast DNA, and investigate the utility for phylogenetic and phylogeographic studies in closely related species of rye genus. Our intention was to select promising candidates of chloroplast DNA regions to guide future phylogenetic and population studies in rye species. Plant material consists of 37 accessions of weedy, wild, and cultivated rye, representing 13 of the most commonly recognized taxa, range, and species of or subspecies. Different numbers of accessions per species were used because of accessibility. Seeds were kindly provided by 3 different germplasm collections from United States, Russia, and from Poland. Our analyses were performed using universal primers for amplifying non-coding spacers of the chloroplast genome. Most of the primers were designed for amplifying spacers between tRNA genes, which provide variety among species or populations. We also used a non-coding spacer region that lies between given genes. 2 genes. This is ribulose biphosphatase carboxylase RBCL and beta subunit of the chloroplast ATP synthetase. After PCR, both strands were sequenced using the same primers as for the amplification. Sequences were aligned and indels were manually coded as a single mutation when they shared identical boundaries and lengths. Nucleotide analyses such as the number of substitutions, indel, parsimoniously informative substitutions, and indels and haplotypes were carried out in DNA as per software. Also, the proportion of variable sites was calculated. We analyzed a total of over 6,400 base pairs obtained from 2016 sequences. The percentage of variability found in each analyzed region ranged from about 1% in ATPB RBCL to 9 over 9% in given intergene spacers. The only source of variability was nucleotide substitution. Two regions, given regions, showed a variation at both population and species level, and two other regions were variable only at the interspecies level. The length of the product obtained correlate in most with those from other species, except for TRNTL non-coding region, which was longer than expected products than in other species. We found a total of 46 haplotypes, most in these two regions, lying between tRNA genes. Based on this data, we selected the most variable regions, which are, of course, the last one, region, and the second one. Two regions, TRNT and L, and TRND and T. These two regions are between tRNA genes, showed variation at both the species and subspecies level, while two other regions were variable only at the interspecific level. We can also see that one region is conservative in related species. As demonstrated in the study, the TRNDT region has shown high occurrence of indels, even in closely related species. TRNLF and DT regions showed hardly any variation, while their adjusted region TRNTL seems to be more informative, as noted by previous studies. The results showed that the investigated regions of chloroplast genome are variable in most of the tested taxa and contain multiple variable regions. These regions should serve as useful molecular markers in phylogenetic studies of closely related species, at least in the interspecific level in Secale genus. In a summary, we can say also that some widely used plastic intergenic spacers can be very conservative in related species. Our data reflects the difficulty of finding the appropriate molecular marker for intra and interspecific studies in closely related plant species. No single region was able to provide a complete taxonomic resolution within the species analyzed when used separately. We can also conclude that some non-coding regions used in the study have potential as a molecular markers in rye genetics and breeding programs. We presume that the analysis of complete chloroplast genome sequences in species of rye will be useful and cost-effective for evolutionary and phylogenetic studies. Therefore, we have started a project of sequencing chloroplast DNA of Secale species. We have sequenced 6 chloroplast genomes so far. One species is already published. This is the most distant species, Secale sylvestre. This is the complete annotated chloroplast genome sequence of Secale sylvestre. It is the first complete chloroplast genome that can be used as a reference genome for further analysis. And at the end, I would like to thank you for inviting me. I would like to thank my team, especially Eva Filip and Izabela Szućko-Kociuba for their excellent work. I would like to thank State Community for Scientific Research for funding this research. And I want to thank you all for your attention.